Chapter 15, Section 2, Breakdown and Synthesis of Glycogen. Now, breakdown of glycogen, because remember glycogen is nothing more than a branched chain polymer of glucose, is referred to as glycogenolysis, where that branched polymer is going to be broken down 1 glucose 1 phosphate by 1 glucose 1 phosphate. The taking of those glucoses and linking them together, that is referred to as glycogenesis, the formation of glycogen. Now when we look at this glycogen breakdown, it's catalyzed by a specific enzyme. This enzyme is known as glycogen phosphorylase. Okay. Glycogen phosphorylase is going to go to the non-reducing end of the glycogen molecule, this sugar polymer. How do I know it's the non-reducing end? Because it is carbon 4 of the terminal sugar. Glycogen phosphorylase is going to take an inorganic phosphate, not ATP, an inorganic phosphate, and it's going to attack the 1,4 branch point. Okay, this glycosidic linkage. It's going to go through, insert, leave hydroxyl, insert the inorganic phosphate. Now you have glucose 1 phosphate that has been released. Now here's the problem though. As I just said, glycogen is a multi-branching polymer. So you have the alpha 1,4 linkages, and then every so often you're going to have an alpha 1,6, where you have a branch point. Okay. The phosphorylase, the glycogen phosphorylase, is going to go after the non-reducing ends, chew up to a certain point that it is now going to cut and transfer. So many units in, it's going to cut from the 1,6 linked sugar, and it's going to transfer to the non-reducing end of one of the strands, and then it's going to keep going and going. Now, glucose 1 phosphate. Glucose 1 phosphate does not appear in glycolysis. So before this can be entered into glycolysis, it has to be altered. Phosphoglucomutase. A mutase is going to take and just rearrange what's already there, which is a bit of a misnomer. Okay? Substrate product will have the same atoms in the molecule. It's just not always the same atoms, that started will be there. You see what I mean here when you look at this figure. Phosphoglucomutase, in its active site, is a serine that has been phosphorylated. G1P enters in. This phosphate group from this serine will attack here at this carbon 6, the 6th carbon. So now you have glucose 1,6 bisphosphate, which actually is the second step, or the third step, sorry, in glycolysis, right before it gets bent, or it could be, depending upon which methods you're using and all, but all right, whatever. So this glucose 1,6 bisphosphate, well, that could be used as is, Problem is, that means phosphoglucomutase is a one-shot pony, a one-trick pony. It can't do anything else because, well, look at the serine. The serine now has a hydroxyl there. What's it to do? Well, it's going to replenish. The phosphate group here on carbon 1 will now be switched up here. So the original incoming inorganic phosphate is now going to remain here on phosphoglucomutase. And what was part of the enzyme, that phosphate group, is now going to leave attached to glucose 6 phosphate, G6P. Now what's going to happen to G6P? Well, depends on, what's the needs of the cell, what's the needs of the tissue, what's the needs of the organism. If you're talking skeletal muscle, because you've broken down the glycogen in your muscles because you're under a high energy needed state, well, G6P automatically going into glycolysis in the cytoplasm. If it's the liver, because remember the liver has the highest glycogen stores, well, G6P is going to be converted into glucose 6-phosphate is going to convert glucose 6-phosphate, G6P, straight into glucose, going to get rid of that inorganic phosphate. And then that glucose is going to be transferred into the bloodstream. So this G6P enters into the ER. In the ER, glucose 6-phosphatase, remember phosphatases remove inorganic phosphate. It's going to remove it, releasing inorganic phosphate, which can then be utilized again. Glucose transferred out. Transporter, GLUT2 is a transporter across the plasma membrane into the bloodstream, carried throughout the body. Now, when it comes time to make glycogen, well, the cells, the liver and the muscle, don't take glucose straight from the bloodstream, add on an inorganic phosphate, and just reverse what we just talked about, linking it together that way. No, there's a few extra steps, okay, a few extra steps to make sure that this is a step, this is a process that needs to be occur, as I've mentioned in lectures many times. Going through and having all these extra steps of complexity gives our cells levels of control. So, complexity here is the fact that you're not just taking glucose, adding a phosphate, G6P, turning it into G1P, and then adding it on, making glycogen. No, you're going to have to go through a sugar nucleotide. A glucose is added as a functional group to the nucleotide UDP, uridine diphosphate. Why do this? Well, it's a limiting step. Okay? Can the sugars be used elsewhere? Can the sugars be sent into glycolysis? Can the sugars, be broken down, sent through glycolysis, utilized either, cellular respiration, pyruvate, then, broken into acetyl-CoA to make fatty acids? Can the parts of the citric acid cycle then be utilized to make amino acids? Blah, 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 blah. If all of those are not required at this moment because the cell, all of its pools, amino acid pools, its fatty acid pools, all of those seem to be where it needs to be, then this process can kick in. Because, to set up that, special UDP we just saw on the last slide, you need to have enzymes be activated. So you're going to have a transcription translational control of whether or not those nucleotides or those enzymes that are going to take and add that sugar on as a functional group to UDP, ADP are even present and active. So you have the sugar phosphate. Okay. You have your nucleotide triphosphate. 1, 2, 3 phosphates, ribose base. So we have here our G1P. Beta gamma phosphates are going to be released. This phosphate, that oxygen is going to attack that phosphate in this phosphate group. So now you have nucleotide dual phosphates. There's the N-diphosphate, NDP, and then the sugar moiety added on there. Where you get the G6P, that's then going to be converted to G1P. Well, hexokinase enzymes can take glucose 6-phosphate from glucose. Add on, say kinase, add on an inorganic phosphate, either taking it directly from ADP or pulling from the pool of inorganic phosphates that are in the cytoplasm or the matrix of the mitochondria. High levels of lactate taken up by the liver, converted to glucose 6-phosphate by gluconeogenesis. So think about it. Your muscles overproduce a lot of lactate. Got to get it out of the body because, can't, for whatever reason, it's not being converted back into pyruvate in the cells that manufactured it. So liver takes it up from the blood. Gluconeogenesis, you get G6P. Phosphoglucomutase that we just talked about, taking G1P, turning it into G6P, also can take G6P, convert it back into G1P, just a complete reverse of what we talked about. UDP glucose pyrophosphorylase takes that G1P that was just made either by hexokinase or lactate gluconeogenesis, phosphoglucomutase turn G6P into G1P, and then UDP glucose pyrophosphorylase takes glucose 1-phosphate, uridine triphosphate, and gives us our UDP glucose. And then it's just a reverse of some of the things we just talked about. This hydroxyl, that inorganic phosphate are going to play well. That oxygen is going to leave that hydroxyl and it's going to bind to this phosphate in this inorganic phosphate, leaving behind that oxygen. That oxygen that is now going to form the glycosidic linkage. And here we have our alpha 1,4. Why alpha? Remember, it's below the plane. Carbon 1, carbon 4. What to do about the branches? Well, glycogen branching enzymes will go through and will go X number of sugar residues from where the branch point, and the branch point is determined by the core. Remember our structure of glycogen. It's going to cut and then shift, and now you have a alpha 1 up here, 6 right there. And you now go from 1 non-reducing to 2 non-reducing ends, which further residues can be added to. Problem is glycogenin. It starts our glycogen strand, our sugar polymer. The thing is, it needs help. It cannot function on its own. It needs a primer of some sort. You see here UDP glucose bringing in the next sugar residues. But where's the things going to be added? How are things going to work here? Well, what we know is that the first reaction is that there's going to be, the sugar is going to be added to a tyrosine residue of glycogenin. Okay? It's not the glycogen core proteins that we talked about last section. Here, the first sugar going to be added directly to the enzyme. The next ones will be added here, boom, boom, boom. And once it grows long enough, this will be cleaved away. And now you have transferred, the sugars will then be transferred to the central structure of the glycogen.